Hello, this is Mitchell's Nerve Project for Miss Meyer's anatomy class. We will be looking at nerve cells, myelinated nerve cells. This means they're wrapped with Schwann cells, which act as insulation. The cell body is where most cell processes occur. It also contains the Nissl bodies and the nucleus, which contains genetic information. We will be following a nerve impulse that begins on one of the branched receptors of the cell, a dendrite. Let us stop at the soma for a moment to consider Nissl bodies. They give gray matter its characteristic color and function like rough ER. They produce vesicles which move along neurofibrils. The importance of this anterograde flow will be seen later. Catching up with our nerve impulse, watch how it moves only between the spaces between the myelin sheath called the nodes of Ranvier. This jumping motion is called saltatory conduction. If the axon, the branched process that carries information away from the cell, is unmyelinated, notice how the action potential moves much more slowly. It also requires more energy. You might be wondering how the action potential moves along the cell membrane. Let's take a closer look to find out. For simplicity, we'll consider one side of this portion of the green axon. From left to right, there are two sodium channels, a potassium channel, and a sodium ion pump. These yellow balls are sodium ions. They're more outside the cell than in. The brown balls are potassium ions. They're more inside the axoplasm. To maintain this concentration gradient, it requires energy. For 1 ATP, 3 sodium ions are moved out, while 2 potassium ions are moved into the cell. Because there are more positive charges outside the cell than in, this results in a net negative resting potential of about negative 70 millivolts. Stimuli may depolarize the membrane, that is, make it more positive. If it reaches a threshold value of negative 55 millivolts, voltage-gated sodium channels will open. This causes the all-or-nothing action potential to occur, and further depolarization as the positive sodium ions move on into the cell. Sodium moves down the concentration gradient into the cell until sodium gates inactivate. Then the potassium gate activates, and this allows positive charge to flow out of the cell, repolarizing the membrane. A dip below resting potential ensures a refractory period during which action potential cannot flow backwards. Let's look at a simple axon and ions to see how the action potential propagates. Stimuli causes the familiar action potential, while ion movement causes a passive current that stimulates nearby membrane to its threshold level, causing its own action potential. How does this work in a myelinated axon? Pretty much the same way, except after the passive current has been formed, it travels a much greater distance and thus saves energy as well as time. Lastly, I will try to explain how an impulse crosses a synapse, the point where two neurons connect. In the left presynaptic nerve are vesicles containing red neurotransmitters. These can be anything from epinephrine to nitrous oxide. On the right postsynaptic nerve are red ion channels controlled by brown neurotransmitter receptors. The question is, how does information cross the synaptic cleft, the space between the two nerves? The synaptic cleft contains calcium ions, which are orange in this case. As the action potential reaches the terminal bulb, it increases its permeability to these ions. The calcium will then begin to enter the presynaptic cell. Doing so causes the vesicles to bond with the cell membrane and perform exocytosis of its neurotransmitters. As a reminder, the synaptic cleft is still filled with sodium ions. Although neurotransmitters enter the synaptic cleft, they do not enter the postsynaptic nerve. Rather, they act upon the receptors on its cell membrane. This can have either an excitatory or an inhibitory effect. In this case, excitatory. This means that the ion channels will become more permeable and move the cell towards its threshold polarization. This will lead to an action potential in the postsynaptic nerve. Sometimes it will take multiple stimuli in the same area or in a small amount of time in order to reach the threshold level. Because neurotransmitters are not used up, they may sometimes be taken back into the cell by endocytosis. This is called reuptake. In fact, some vesicles may even move back into the cell body for processing. This is called retrograde exoplasmic flow.